Hello and welcome to my 8th video on IB Psychology. Today we're talking about the biological perspective. My name's Linnea Karlberg and I'm an IB 12 student. We're going to keep going where we were, talking about genetics. Genetic relatedness refers to the extent to which two individuals have a genetic similarity. And then through this, correlation can be established between similarities and behavior. So normal siblings have a relatedness of 50%. But monozygotic twins, which we can see here, have a genetic relatedness of 100%, so all the same genes. Through this knowledge, twin studies are performed, which compare behavioral traits of monozygotic twins and dizygotic twins to evaluate the degree of genetic and environmental influence on a specific trait. And these are used to operate under the assumption that any major difference in behavior, a disease, or physiological trait that appeared in one twin is a result of environmental factors, thus low heritability. And if the trait was a result of genetic factors, then both twins would manifest the trait. However, epigenetics has changed this assumption and created a more complex and nuanced approach to monozygotic twin research. So what I was talking about is this equal environment assumption that twins are raised in the same culture by the same parents and the same surroundings. Thus, it's environmental factors affecting genetic expression or behavior development. They should be equal. But that ignores the environment in a statistical calculation, meaning that the variable is the same for both twins. And here a trait is any physical or behavioral expression of a gene, and genetics attempt to isolate certain traits and identify the gene responsible for the creation of that trait. The concordance rate is then the extent to which a given trait is shared by a pair of individuals expressed as a percentage. So yeah, this assumption is often erroneous because twins tend to experience differences in the environment that can have significant effects on physiology and behavior, like nutrition, peer groups, treatment by parents, and stress levels. So what I was talking about the epigenetics here that affect the monozygotic twins is here we can follow Jennifer and Karen. They're identical twins born with genes that absorb fats twice as fast as the average person. However, they make different life choices. Different environmental factors come into play here through their diet. Jennifer eats healthy low-fat foods, so her obesity genes are not triggered. And Karen eats fatty and healthy foods, so her obesity genes are triggered. Now that we can evaluate these twin studies, let's look into two examples. First, there's Bouchard et al. In 1990, who looked into does monozygotic twins have similarity personality traits even when separated? So this was a longitudinal study. It's been ongoing since 1979 at the University of Minnesota. And the aim is to track down separated twins from across the world, and then the participants complete approximately 50 hours of medical psychological assessments, looking at their personality traits, occupational interests, and mental ability. And it was found that an identical twin reared away from his or her co-twin seems to have about an equal chance of being similar to the co-twin in terms of personality, interests, and attitudes, as one who has been reared with his or her co-twin. So the similarities then between twins are due to genetics, supposedly, and the differences between twins reared apart must mainly be due to environments. Candler et al. In 2006 looked into a twin study studying determining the level of irritability of depression. And we're going to talk about the irritability of depression as well later on. So over 42,000 twins from the Swedish National Twin Registry were sampled for the study, and the average concordance rates for monozygotic male twins was 31%, and the monozygotic female twins was 44%, versus then in the dizygotic twins, they have the same genetic irritability as siblings, was 11% and 16%. So the irritability of depression is estimated to be roughly 38%. So depression may be the result of a genetic predisposition. However, it does not contradict the argument that depression is genetically inherited. But the gene may be there, but lived in a different environmental conditions, such as one with more stress, and thus have not expressed their genes. And in some cases, the dizygotic twins both had depression, and we need to remember that they share some of the same genes. Overall, this has inspired the Human Genome Project, which is a genetic mapping that showed that up to 11 genetic markers variations that seems to be correlated with major depressive disorder. There are also a lot of kinship studies that are done to learn more about genetics. So, for instance, family studies, which trace a phenotype over several generations in a family tree, and then attempt to determine the likelihood that a behavior is inherited. And there's also adoption studies, which investigate similarities between the adoptee and their biological and adoptive parents. And they look into the similarity with the biological and see that as a result of genetic inheritance and similarities with the adoptive parents, and assume that's a result of environmental factors. Once again, epigenetics changes all of this and makes it a bit more complicated. Heritability is a statistic used to measure how much of the variation in a trait is due to variation in genes, and is usually reported as a value between 0 and 1, and is often converted to a percentage. Heritable means differences in a trait from one person to the next are explained to a certain extent by genetic differences. So the higher heritability, the variation in the trait is due to genetics. However, in often cases just estimates. We're not able to fully integrate the complexity of genetic interactions that we know occur. Once again, epigenetic factors of gene expression and repression. Also, this doesn't provide insight into specific genetic functioning, and it may create a misleading belief in DNA as determinism. Now let's bring it back to a study about depression once again. Capps et al. In 2003 looked into the role of the 5-HTT gene in depression. And this gene is known to play a role in serotonin pathways, involved in controlling mood, emotions, aggression, sleep, and anxiety. So the hypothesis was that people who inherit too short version of the 5-HTT gene are more likely to develop major depression after a stressful event. This was a longitudinal study which sampled 847 New Zealand 26-years-old who had all been assessed for mental health on an every other year basis until they were 21. They were divided into 3 groups based on their 5-HTT alleles. Group 1 had short alleles, Group 2 had 1 short and 1 long allele, and Group 3 had 2 long alleles. And the mutation of the 5-HTT gene has the shortest alleles. The participants then performed a stressful life events questionnaire, which questioned them the frequency of 14 different events, like financial, employment, health, and relationship stressors between the ages of 21 and 26. And the participants were also assessed for depression, as I mentioned earlier. And it was found that people who had inherited one or more short version of the allele demonstrated more symptoms of depression and suicidal ideation in response to stressful life events. So the effect was strongest for those with 3 or more stressful life events. So it's important to note here that inheriting the gene was not enough to lead to depression. The gene's interaction with stressful life events increased one's likelihood of developing depression. That's what activates the gene. So this study, looking into strength, it acknowledges the interaction between biological and environmental factors in depression. So it takes a very holistic approach in contrast to a reductionist one. However, important to note here, this was a correlational study, so we cannot make a cause and effect relationship. Also, it operates under the assumption that serotonin causes depression. And can we really trust the self-reports? Also, the salience of the negative life event which plays a role in depression. It's possible that those that recall their stressful life events more easily may have a tendency towards depression, not those who have experienced them. Some people may be resilient and not recall negative life events easily. And also, some participants who didn't carry the gene mutation became depressed. So we can't say that the gene expression alone can cause depression. Waterline and Girdle et al. In 2003 looked into the role of nutrition in epigenetic processes. They obtained infant mice that were the offspring of over 200 generations of Agouti mice breeding. And the expression of the Agouti gene results in two distinct phenotypes, yellow fur and obesity. At 8 weeks of age, these mice were randomly assigned condition: the control condition, which were fed a normal diet of mouse food in pellet form, or the experimental condition, which were fed a normal diet plus methyl-enriched pellets. This diet was for 2 weeks prior to mating with other Agouti males and during pregnancy and lactation. It was found that the Agouti mothers in the experimental condition gave birth to brown mice who, monitored into adulthood, maintained a normal weight and lifespan, in contrast to mice with an expressed Agouti gene who died prematurely due to their obesity. So, genetic expression could be altered simply by a change of diet. This study shows strong support for epigenetics in action, and this was a controlled lab experiment, so we can see a cause and effect relationship. It also sparks future research into developing epigenetic interventions in order to treat diseases. However, creating methyl-rich diets in order to silence genes detrimental to our existence, it's not easy. We have no idea what other genes were methylized as a result of the methyl-enriched diet, so maybe this would be dangerous to perform with humans. And once again, if you're an HL, you can talk a lot about here what it's like to research with animals, the strengths and limitations of that, which you will have an extension on. Thank you so much for listening. Let me know if you have any questions.